This is the 2026 WAEC Biology Practical, and I'm here to give a prediction of the kind of questions you're going to expect. Okay, so I'm very confident that at least up to, let me say, 80% of the questions here will come outside in the exam, or maybe 70%. But at least I'll just give you an idea of, okay, read this part, learn this diagram, or okay, the question can come this way. I'll just give you the idea in this video. Okay, so these are the list of specimen. So here we have your granules. So this is a granite pod. You have a crushed granules. This specimen is B, and you have your water, and you have a salt solution. So for this water and salt solution, this specimen C and D. So one thing you need to understand is your mind should go to osmosis and diffusion under the types of solution. So we have hypotonic solution, or have hypertonic solution. Okay, so you just need to revise and go back to that topic so you can know how to prepare for a question like this. Because for you to have just water, which can be distilled water, and a salt solution, so you can ask you explain the type of solution. All right, so that is very important. So now we have sandy soil with the specimen E. Have sandy soil. You have mammal digestive tract, which is from the esophagus down to the stomach. Okay, then we have the mistletoe on the host plants. Okay, these are those unwanted plants that is found attached to other plants. They use the plant for support, and what they do, they derive nutrient from the plant. So this is a very good example, a mistletoe. Then you have your specimen H. You have your milk. Then you have your fresh fish. Yeah. So for the next specimen, specimen K, you have your maize grain. You have your tomato paste. You have a fresh tomato. You have your rotten tomato. So with this, they will always ask you which organism causes this type of damage to the tomato. So it's a fungi. Okay. Then thermometer, that's for ecology. Metre, that's for ecologist. Then you have a quadrant used for the quadrant. There's a quadrant here, yeah. Used for population studies to know the number of species present in a particular habitat. So okay, so I'm going to go through the required materials. So we have a hand lens and magnifying lens, very important. Then you have your beaker. You can't do without beaker in scientific and experiment. You have a freshly prepared iodine. So for you to see iodine, your mind should go to test for starch, which is very important. You must use iodine. Then you have your sodium hydroxide solution. Then you have your copper 2 sulfate, your white tile, you have your Benedict solution, and you have your filter paper. Okay, so these are the materials required. All right, so the first thing we're going to do, we'll just go through the food test. All right, so carry out appropriate food test on specimen B, which is cross granules. You have your H, which is milk. Then you have your maize grain to determine the presence of the following. So here we have test for starch. You have a protein. Test for starch, protein, reducing sugar and oil, sugar fat. So for test for starch, it's very straightforward. All you need is your iodine solution. You just put a drop of this iodine solution in this specimen and see which of them is going to give you a bluish black coloration, which is evidence that the starch present. So if you put this drop in a crushed granite, there will not be any color change. You put this drop in the milk, there will not be any color change because your granite and the milk, they're both protein, so they don't contain starch. But if you put this in your maize grain, it's going to give you this bluish black coloration to show that starch is what's present. Okay, so this is one thing you need to understand. So this is a very straightforward experiment. Sometimes you don't actually need to carry out the experiments, you just need to know it straight away to know that okay, maize grain contains starch, and when you add iodine solution to it, it's going to give you a bluish black coloration. So that is one. Then the next one, we'll have a test for reducing sugar. Okay, and this is a Benedict solution. All right, so this is milk. If you add it in a test tube, don't mind this is just a pot, but normally we use a beaker. All right, we use the beaker for this experiment instead of the pot. So use beaker in place of this. So in the boiling beaker, you place the test tube of the milk. Then what's going to happen next is color change. So there will be a difference in the color. After applying heat to this solution. So we're going to have different color change. Okay. So what we're going to see here, you can see it can change from blue to green to yellow and to brick red. All right. So this different color change occurs, and I'm going to tell you why we have different color change in this solution. Now, now see this. So procedure: put a small amount of milk into a test tube, and you add a Benedict solution, and you heat the boiling water bath for 2 to 3 minutes. So when you heat it, what you're going to get is a different color change. All right, so the color changes from blue to green, yellow, then the brick red precipitate. So this brick red precipitate shows an evidence of you reducing sugar. So most experiments, most things you check is the color changes that occur for you to be able to detect that okay, this is what I'm looking for and this is what it is. Okay, so the inference is within sugar is present, and why is heating necessary? So heat provide energy to reduce copper 2 ions to copper 1 oxide, forming the colored precipitate, which is the brick red color we're able to observe. Okay. I doubt if they're going to ask you this, but in case they ask you this is the answer to that question. Okay, so test for protein, and this is the Buret test. So procedure, you add sodium hydroxide solution to the specimen extract, add 2 to 3 drops of copper sulfate solution. So you can stir it, keep stirring. When you add the drop, you stir it and wait for the color change. Just like if you're doing your titration. So you add a drop and check it, then there'll be a color change. Okay, so for this, you're supposed to know that yeah, granules contains protein. So if you put it on granite solution, you have a violet to purple color. Then if you put this in milk, it give you also a violet color also. Okay, so it's very important that granules and milk, they both contains protein. So when you add that solution to both of them, it gives you this violet color to show that protein is present. So very important. Okay, so the next one, yeah, inference protein is present. So the next one is test for fat and oil. So the reason why I say fat and oil, I know in the experiment, you're not asked to bring oil, but I just assume that okay, because you have a crushed granite and granite contains oil, they may ask you to crush the granite, place it in a test tube, or place it in the filter paper. You rub it, and this is what you're going to get. You're going to get a kind of translucent surface like this. And when something is translucent, it means that they allow light to pass through, but not always clearly enough to see details or images through them. All right. So oil is translucent. So if you put it close to the sun, you can see a little bit of the image, but light cannot usually pass through it very, very well. So this is one of the evidence to show that okay, this contains fat and oil. I think this question is very common. I think last year and other years, this is very common in biology practical. So just check out for this, it's very important. Then the next one, this is just a summary table. You go through this table, all right? It gives you everything about the experiment. So you have your specimen here, you have your specimen, you have your test, you have the observation, and you have your inference. So all these things here are very important. So you just check the overview, and if you look at it, color present, purple color, black brick red precipitated, blue black coloration, translucent spot formed. Color observation, it gives you an overview that okay, this is starch, protein, or whatnot, fat and oil. Okay, so let's go through this table and try to make sense out of it. So for soil particle, for you to have sandy soil, it's very important that yeah, they're going to ask you major question on this. So place E is sandy soil in specimen C, which is water. So you can tell you to put water into the sandy soil, and state observation. Sandy soil have lots of particles, and during rainy season, if you notice, you're observant, you see that you start seeing bubbles of water coming out of the soil. Why? Because sandy soil have a lot of air and is well aerated, so you start getting bubbles coming out like this from it. Okay, so having bubbles coming out of it. All right. So this is just one evidence that okay, this soil is quite sandy. All right, because of the air which is highly present in sandy soil. Okay, so soil settles quickly. It settles quickly because the sandy soil has loose particles, so it actually settles quickly. Then the water becomes slightly cloudy. Once you put it, it becomes cloudy. The water becomes very cloudy because it contains a lot of dust, so you cannot see through it. It's not transparent very well. So because of the presence of dust particles found in the water, so it becomes very cloudy. Then you have large particle visible. Sometimes you can have some big stones in sandy soil, which is very visible. Okay, just like this. So in the sandy soil, you can see some stones, some particles around it, and it's very visible. So this is very good evidence that this is a sandy soil. So if we state four characteristics of sandy soil, all right. So with this, it's very important for you to know that sandy soil have large particles that are well aerated. They have a lot of air present. Then it drains water quickly. So once you put water in sandy soil, the water pass through it very fast. So it drains it very quick. Then what low water retention means sandy soil cannot hold water for a very long time. That's why it's not really good crop for agriculture. Okay. So once you pour water, it drains very quick. So it retains water very low. So you have a low water retentive capacity. So these are just so you can check for other characteristics, other characteristics of sandy soil. Agriculture will tell you okay that sandy soil is have loose particles, then they are very sticky to the touch, very sticky to the touch. So these are just kind of some characteristics of it. Then state to advantage of sandy soil. So advantage, good drainage, easy root penetration. Okay. Then the next one, two disadvantages of sandy soil. So the poor nutrient retention, they don't really contain much nutrient because they are highly aerated and they can drain very quick. So they don't really contain a lot of nutrient like that. And again, they dries very quick. Okay. So mention three crops grown in sandy soil. So it's very important. I'm very sure they can ask you those questions. So have your groundnut, have your cassava, and have a watermelon. So these are just a few of crops that can grow very well in sandy soil. Trust me, they're not going to ask you, why do you choose this crop? They're not going to ask you those questions at all. So the next one, okay, it tell you to identify specimen P, Q, and R. So these specimens for P is thermometer, Q is a meter rule, and R is quadrant. So for this, they can just ask you, okay, state one use of each of them. And that's straightforward, that yeah, thermometer measures temperature, and meter rule measures distance or length, and the quadrant estimates population density. Okay, so these are something we need to take into consideration. So population density, the amount of organism present in a particular area. Okay, so very very important. Then how explain how a quadrant is used. So they can just ask you, okay, you throw the quadrants randomly, you throw it randomly in the habitat. You don't use a specific way, just throw it randomly. Then you count the amount of organism within the quadrant. So the quadrant is kind of rectangular. It has a lot of boxes. So you count the organisms within that quadrant. Then you have to repeat this several times and calculate the average population. Okay. So for example, this is the quadrant here. So you count all the organisms present in these boxes here, all these boxes here. You count this organism. So in the real sense, for example, the organism we found here can be the same organism we found here, or you can find a different organism here. Okay, but they will not ask you that deep technical question on this, because if they want to go deep on this, then you just more asking a typical ecological question which is above the scope of WAEC. Okay, so this is the quadrant. These are people studying quadrants. This is another person studying quadrants. So with this, if you look at this expected observation table, now this is your first row. You chose the first time, you chose second time, third time, fourth time, and fifth time. So number of plant counted for each row. So here is 8, 11, 9, 12, and 10. So these are the number of plants counted. So this is just kind of a summary. So if you add everything together, the number counted, this is total count. You have a 50. The number of throws, how many times do you throw the quadrant? Is 5 times. Then average count. So you divide 50 by 5 and you get what's 10. So it's a very straightforward calculation and very easy to understand. Okay, so the next one, state three precaution when using the quadrant. So number one, throw randomly. The first one, they avoid counting outside organisms. So count within the box and take many readings to get an accurate readings. Okay, so that is that. So identification of specimen A, this is the groundnut pod. And one thing you need to know about groundnut is that groundnuts are cover crops. Okay, they're cover crops, and we have other species of groundnuts that are cover crops. Okay, so we have a species called I think Pride of Barbados. All right, Pride of Barbados is a very good example of a cover crop. So and again, what type of fruit do you think groundnut is? All right, so it's a legume. So the tissue classification of crops. So this is what a legume. All right, and you can see how the picture look like here. It has have a lot of pods. So call this a pod, and this is the Pride of Barbados pod I was talking about. So you can see that they belong to the same family. All right, so you can see the pod. This makes you understand that okay, these are all pod bearing fruits. All right, and they're known as what the cover crops. Okay, so state four observable features of this granite pod. The brown in color is a dry fruit. Ask you what type of fruit is this? It's a dry fruit. And in classification of fruit, we have what we call dry and dehiscence and dry dehiscence. Dehiscence means break open. So granite pod normally break open, so it's a dry dehiscence fruit. You have a rough outer surface and it contains seeds. When you break it open, you have some seeds inside. Okay, so you break it open, you have some seeds. So state four use of specimen B. All right, so we have the crushed granite. All right, and the crushed granite is very important. So when you crush the granite, for example, you can use it for a lot of things. So you can use it to produce cooking oil. You can use it for human food, livestock feed, and production of peanut butter. Okay, so this is crushed granite. This is not the granite pod. This is crushed granite. Okay, so this is what you can use it for, and it's very good for livestock. Then you have the alimentary canal, which is the tube that connects your gullet to the stomach, the long tube, which helps in digestion. So when you eat a food, you swallow it, pass through that tube, and get to your stomach so that the food can be digested properly. So specimen F is what mammalian alimentary canal. So what you need to do here, you need to learn how to draw the diagram. I'm very sure to ask you to draw the diagram of this. So I'm just using a goat for example. They didn't indicate whether it's a ruminant animal or a non-ruminant animal. So for here, yeah, ruminant animal in the stomach they have the reticulum, omasum, and bomasum, and then you have the rumen. All right. So they won't ask you. I'm very sure they will not ask you to explain the complex stomach of the ruminant animal. So start explaining rumen, reticulum, I'm not sure. But the thing they ask you to do is to draw the diagram. You draw the diagram and they tell you, okay, what the function of the alimentary canal, okay, what the function of the stomach and stuff like that. So just to go through this, 64 function of the stomach, then you have your temporary storage of food, digestion of proteins, churning of food to break down the food into smaller particles, destruction of every harmful microorganisms. Then stage two digestive enzymes found in the stomach. So you have your pepsin, you have your renin, especially in your young mammals. So apart from the stomach, the other aspect of the alimentary canal that you need to learn. So what you can do is you draw the diagram of the alimentary canal and try to know the function of those parts. So you can get an overview of these if you check digestive systems in biology. So if you check it, you can get a diagram of that and you see the part of it. I don't want to go way overboard during the diagram, start going through the um the function of each part. It's going to make the video very long. So I'm trying to give you a reference of what you can do. State for adaptations of the stomach. So here, thick muscular walls, gastric gland present. Then you have expandable walls, and production of hydrochloric acid. So hydrochloric acid is very important in the stomach because it helps in digestion and it helps to kill some microorganisms that's not good in the body. Then identify specimen G. Specimen G is the Mistletoe, which is the plant that grows on other plants and serve as a parasite. Okay, this is a Mistletoe, and this is another Mistletoe. We have different species of Mistletoe present. All right. So state the biological relationship. We have different relationship in biology. So this is parasitism because one organism tends to derive nutrient from the other organism, and the other organism does not benefit at all from this type of what association. So this is parasitism. So explain how mistletoe obtains its food. So mistletoe, the scent sucking root called the hostoria. Okay, the hostoria stick to the stem and they tend to what, to absorb water and nutrients from the stem. You know, most plants they use their roots to absorb water. So the mistletoe will just be on the stem waiting. So when that nutrient or water is coming, they tend to suck from the stem from the nutrients that the plant are already absorbed into the stem. Okay, so this is an hostoria of the mistletoe. All right. So state four harmful effects on the hosts. What they do is they reduce the growth of the plant, loss of nutrients, reduce yield, and death of branches. Those branches tend to die off because they're not getting enough nutrients for them to remain stem and firm. Okay, so mention two other plant parasites. It's very important for you to know other plant parasites that can actually cause this type of situation. So you have a doda plant, also known as coscuta. So this is the doda plant. So you can see how it looks like. They have a witchweed, also known as a striga. So this is a witchweed. They are so very parasitic. Okay, so identify specimen J. Specimen J is the fish, and you know that it is very mandatory that you should know how to draw the fish and label the fish. And in this video, I'm not going to tell you the function of the pectoral fin, the dorsal fin, the caudal fin, the mouth, the gill flap, the pelvic, and everything. You need to know how to draw it, label it, and know the function of all the parts. It's very, very important to learn that yourself. Okay, so state four observable features of the fish. The streamlined body, they made them to swim very well. Streamlined body, pairs of scales, pairs of fins for swimming, and pairs of gills for breathing. So it's very important to get all these ones. Then state for adaptations for aquatic life. And the first thing you need to know, what I didn't mention here in the video, what the class, which class does fish belong to? They become a Pisces. Pisces, okay, that is the class of fish, and the phylum, the other Animalia, okay, and their habitat is what aquatic. So you have to take note of this information. It's very, very important. So, six for adaptations for aquatic life. The house streamlined body reduces water resistance, fins aid movement, gills enable respiration, and scales protect the body. They're very important. Then, state for economic importance of fish. What do you use fish for? Now, source of protein, very important. Source of income. When you sell them in the market, you use the income to survive. Okay. Export commodity for import and export. Raw materials for fish meal. So you could create meals, fish meal for your livestock or for your family. So these are just the importance of fish. And tomato study. So the next thing here we need to learn about tomatoes here is how I just want to give you a table that give you an overview of both specimen M and N. So we have your fresh tomato, we have your rotten tomatoes. So for both the color, we have the bright red, and for the rotten one, you have a dark red, brown color. Texture, firm for the fresh tomatoes, and you have soft for the rotten tomatoes. You talk about the physical features of both of them. Then the smell, fresh smell from the fresh one, and the other one is offensive smell. While the appearance for the fresh one is smooth, while for the rotten one is what's wrinkled. Okay, so state four causes of spoilage. So it's very important. Fungi, bacteria, high temperature, and physical injury. So if it pierces the tomatoes, what happens? They become exposed to bacteria and fungi, and they can still come inside and cause infection on it. So this is one thing you need to understand. They won't ask you give the name of the fungi that causes this infection. They won't ask you that, but you don't need to know that the causative organism is was fungus. So it's very important. So state form method of preservation. So this is very important. Okay, how can you preserve tomatoes? Refrigeration, drying, canning. Okay, So canning is very important. So you put them in cans, put them in cans to make them to be viable for a very long time. Then you process them into paste. Okay, process the paste to use in cooking and all that. So these are those methods of preservation of tomatoes. Then, yeah, just a comparison between tomato paste, which is specimen L. Tomato paste, if you refer to the first to the beginning of the lesson, I give you a picture how it looks like. Then we start with fresh tomatoes. Then the form, processed and unprocessed. Tomato paste is already processed already, while the fresh tomato is unprocessed and not reprocessed at all. Then the texture, tomato paste is a thick paste, is very thick, while fresh is what firm fruit. Water content lower, higher. Shell life, though I'm quite skeptical if they will ask you something like this to compare tomato paste and that of fresh tomatoes. But that tomato paste is used in industry. They is used in industry, and is also used at home to cook different type of meal. Okay, so it's very important that you just have get more information on what this tomato paste is all about. So this is just kind of the comparison between tomato paste and fresh tomatoes. All right, so you just go through it, just play with it in case. But I'm kind of 60% or 70%, let me say 50% sure that they'll bring something like this in this exam. But I just put it here in case it's of any help to someone that actually need it. Okay, so one important thing you need to understand here is that you need to learn some specific drawings, which is very compulsory. That yeah, these drawings are going to come out in the examination. All right. So this granite pod, I doubt it's going to come out. Granite seed, I doubt they tell you to draw granite seed, but fish is 100% certain that it's going to come out. They're going to draw fish, label the fish, and write the function of those parts of label. Mammalian Alimentary Canal, I'm very certain that they will tell you to draw the alimentary canal of mammals and tell you to describe it. They'll tell you that okay, what the function of the alimentary canal and all those things. So those refer back to digestive system and try to get more overview on that. So you can look for agricultural textbook and and get more overview on just ruminant and non-ruminant animals and see what you can get from there. So for this this tomato fresh fruit, I don't know. They can tell you to get a knife and split it into two, and they may ask you a question like they'll tell you, okay, draw this part of it and label it and tell you, okay, what type of placentation is this? And this is axial. This is axial placentation. So it's very important for you to get this info. So for Mistletoe on host plant, they cannot tell you to draw Mistletoe. It doesn't make any sense. They will tell you to draw it, and they cannot tell you to draw the quadrants. It doesn't make any sense. Just drawing a triangular box or rectangular box rather, and they can't tell you that because it doesn't make any sense. So you have to take note of these three important things: fish, mammalian stomach and musicana, and fresh tomato fruits. So it's very important that you learn all this for the exam. So this is just my take and this is just my ideology that okay, this all these questions can come outside and it can help you in preparing for the exam. All right. So if you have any question, you ask me this question in the comment section, and I'll try my best to answer everything before your examination. All right. So after your examination, you can come back here and check it and say, oh yeah, most of the questions, most of the things you touch here actually come outside. All right. And these are the few that didn't come outside. And I can tell you that okay, this is what you wrote here is correct, or you can just give me an overview that okay, these are the questions that came outside on your slide. All right, and I'll be able to go through it and tell you that okay, yeah, this is how it's supposed to be and this is the correct answer. All right, so this is the end of this video, and I believe that my information here was quite helpful for you preparing for this exam, and I'm going to see you next time. But please subscribe to my channel so I can get the latest of my video. Don't forget that my aim is to teach all topics in biology, and I'm here to make it more interesting so that when you learn my topic, when you learn my teaching, you'll be able to understand the basic, and biology will become very easy and more simplified for you. Okay, so see you next time, and goodbye.